So, talking to our audience and our remote audience. So this is a little odd for us. We're going to give it a try. But if you got this week's Western Farm Press, and I will hold that up here, perhaps you can read this online. You'll see that the title says, "In California, cotton seeking to get upper hand on whitefly." Okay, that's an important issue for California cotton, and any Arizona grower knows that's really important in Arizona too. But why is it making the front page of Western Farm Press this year? And that is handled in the secondary headline, which is on the inside cover, and it says, I hope you can read it, "Sticky cotton, a concern for California growers." Well, I guess being from Arizona, I'm real glad it says California growers, because we don't even like the word stickiness being mentioned in the same breath with Arizona cotton. But the fact is, they're talking about it very openly in California, because there's been a few incidents from last year's crop from California that got flagged as sticky by buyers, and that is not something we can sustain in our industry and expect to maintain any kind of premium or, in fact, any kind of industry for cotton over time. So managing whiteflies is critically important. I've talked about it many, many times, and I've presented the fundamentals. All of our guidelines are available online. But today, I thought it would be good, given the interest in whiteflies and whitefly management currently, to talk about some of our newer procedures. They're just being developed by a graduate student, Tim Vandervoot, who's going to talk about integrating natural enemies into the management of whiteflies in cotton, as you can see there on the title. This is particularly important in an era where we're reconsidering the use of broad-spectrum chemistry for the control of brown stink bug or other stink bug pests that enter our fields this year. Stink bugs have been an issue the last couple of years, and it's something that our growers need to grapple with. But part of that decision-making needs to be, what are the impacts on whitefly management? So I'll turn it over to Tim, and he'll talk about that topic.
Good morning. Thank you for attending remotely and in person. My name is Tim Vandervoot. I'm a graduate student at the University of Arizona, and like Dr. Ellsworth said, we've been doing some research, and we're very interested in the role of natural enemies managing and suppressing whiteflies in cotton. So just to start off, we talk about natural enemies, and I think a lot of us are very familiar with them as agents that play a positive role in cotton. But I think in the past it's been more of a passive recognition. Now I'm interested in everyone's awareness and knowledge of natural enemies. Through my research, or our research, we've focused in on a few that are very important. I have some pictures up here of a few natural enemies. I guess I'll just talk through them real briefly. I want to reinforce the idea that it's important for everyone to be able to identify what is a natural enemy and what's a pest. Even if you rely upon your PCA to make a spray decision, these are all very useful agents, and it's to your credit to be able to know what's going on in your field, in your system. So this is a picture of my new pirate bug, an adult, and it's nymph. These are very important predators in the cotton system. They're piercing, sucking predators. They feed on whitefly large nymphs. What we have here is a big-eyed bug. Again, very, very similar feeding habit to the minute pirate bug. These are kind of a workhorse of our natural enemy community. They consume a lot of whiteflies, and we've identified them in our research as being another important predator. Not an insect, but a spider, an arthropod. A crab spider is very important. In this picture, it's feeding on Lygus nymph. But these spiders also eat a lot of whitefly adults. Another important predator. I should point out that a lot of spiders are very important in our system. but we're focusing on crab spider because it's so closely correlated to whitefly populations. And finally, the Colops, soft-bodied beetle. This also feeds on the whitefly large nymph, but it's a chewing predator. If anyone goes out and looks at their cotton and looks at the cotton leaf and sees a cadaver of whiteflies, indication that this has been preying on your whiteflies is that those large nymphs have been chewed upon, not just evacuated, but partially destroyed. So that's just a little bit of background of natural enemies that are important in our system. I'm going to tell you why and how we reach these conclusions. I guess I could back up a little bit. We talk about natural enemies being important in whitefly management, but really we should talk about why whiteflies are important. We have a very good IPM system in place that can manage whiteflies, but in a lot of scenarios, whiteflies can exceed the boundaries of control and cause secondary pest outbreaks. Here's a good example of a field that Dr. Ellsworth mentioned earlier. We pulled this out of a field in Blythe at the end of last year. This is very productive cotton, but it is being recognized that it can be, even in the most productive field, you can have a severe whitefly infestation that can really limit the quality of the lint that you're producing. And this is the same branch, just turned over. On one side, heavy infestation with sooty mold as a result of honeydew being excreted by whiteflies. What caused this whitefly outbreak is probably brown stink bug management, aggressive brown stink bug management. Unfortunately, the chemistry we have for brown stink bug control is broad spectrum at this point in time, and those broad spectrum insecticides really knock down our natural enemy community. So it's important to know what our natural enemy community is doing, just so that we can minimize situations like this and maintain the quality of our fiber. Now, we have an excellent measurement system for whiteflies. Hopefully, we can prevent those types of situations. It relies upon active sampling, turning the fifth main stem leaf, looking for adults, and also looking on the leaf disc for large nymphs. And with that sampling, we can make decisions about the level of infestation in our cotton system. Now, there are situations where it's very easy to make a decision based off of the sampling, and situations where it can be quite difficult to make a decision. We look at this table, when you have low infestations, 0 to 25%, it should be very clear that no spray is necessary, and high infestations, you're certainly possibly behind the curve, where you need to be making a spray immediately. But we have scenarios within this decision-making matrix that are uncertain. We talk about our threshold being around 40% infestation of leaves with adults, 3 or more adults, and 40% infestation of leaf discs with large nymphs. But that threshold is not a clear line. We shouldn't consider it as being, prior to 40 you don't spray and after 40 you do. It's actually a zone from 25% to 52% infestation. And that's where your PCA's knowledge and background and experience come into play in interpreting these populations and making spray decisions. So, I talked about the importance of natural enemies in suppressing whiteflies. They're also useful, we think, in minimizing the uncertainty around these areas. That they can be another tool in making spray decisions. But we do have a lot of natural enemies in our system. have over 20 arthropod predators, spiders and insects that prey upon whiteflies and parasitize them. And these predators and parasitoids vary from year to year and throughout the season in terms of importance in suppressing whiteflies. In fact, one year you might see the dominance of one species or genus, and the next year a different cohort suppressing whiteflies. So part of our research was to identify natural enemies that play an important role in suppressing whiteflies and can be used as reliable indicators of control throughout the season. Now, I'll spend just a real brief moment on this. This is just to tell you or kind of explain the relationships that we're talking about when we talk about natural enemies and whiteflies. What we have here is data that we collected over the past 2 years or 3 years at Maricopa Ag Center. We're looking at whitefly natural enemy interactions under a variety of treatments. And each point in the graph is representation of different whitefly and predator levels under different treatments. And we make spray decisions based off of our whitefly populations presently. We talk about 40% infestation, that's equivalent to 1 large nymph per disc. So if we weren't considering our natural enemies, we would just say below 1 or actually below 0.5 large nymphs per disc. Would not make a spray decision, and above 1.5 we would. But you can see how this relates to natural enemies. Below 0.5 large nymphs per disc, we have very high natural enemy or essentially predators on this x-axis to whiteflies. And above 1.5 large nymphs per disc, we have very low natural enemy ratios to whiteflies. In between, we have that zone of uncertainty from 0.5 to 1.5 large nymphs per disc. That's equivalent to 26 to 52% infestation. So we really want to help make decisions around this threshold a little bit more clear. And using the natural enemy populations or the ratio of natural enemies to whiteflies, we think we can do that. So instead of making a spray decision around our current conventional threshold, we think we can do that with our predator to whitefly ratios. What we see is a relationship just as well on the y-axis where ratios of predator to prey above a certain level indicate control. Those whiteflies are being suppressed. And below this ratio, a lack of control or a lack of suppression. So I mentioned earlier 4 natural enemies that are important in our system. It's in fact these 4 that we're focusing on as indicators of biocontrol or suppression of whiteflies in our system. With big-eyed bug, pirate bug, coleops, and crab spider. And we chose this out of the research that we did. We chose these 4 predators as being the most useful indicators of biocontrol because, one, they're very closely correlated to whitefly populations. We have a lot of predators in our system, but for a lot of different reasons, they don't necessarily track with whitefly populations. And also these 4 predators are reliable from year to year and all season long. And there's some variation in the course of the season, but what we're looking for in this research is predators that will indicate the natural enemy community's effect on whiteflies or the suppression of whiteflies. So that's what these 4 are. They're indicators of the entire complex of natural enemies. Now we don't want to add additional work to PCA's sampling, so we tried to come up with a way to measure the effect of these natural enemies at any given time in a cotton field. We found that sweeps were the most effective, which is fortunate because I think a lot of PCAs or all PCAs should be taking sweeps, certainly if you're looking for ligas. And the relationship of, in this case, Aureus or pirate bug as sampled by sweeps, can be compared to whitefly large nymphs. Like I said earlier, Orius are very closely, they're predators of whitefly large nymphs, are piercing sucking predators. So, sampling out of 100 sweeps, if you found 5 minute pirate bugs, and you had that to compare to a population of whiteflies that were represented by an average of 1 large nymph per disc, which is an equivalent to 40% infested, we would say that ratio is indicating control or suppression of whiteflies. So we've another way to look at this is if you're taking 100 sweeps in your field and you're finding a lot of minute pirate bugs, certainly more than 5, and you have an average of 1 large nymph or fewer, then control would be exhibited. The ratio is a little bit tricky to think about off the top of your head, so we built some tables that help identify this a little bit better. We have a few of them here, but essentially what they are whitefly population comparisons to the predator that corresponds to them. In this column here, I don't know if you can see it, in the middle here we have the number of leaf discs infested with large nymphs. It's 1 or more large nymphs per disc. How that translates to percent infested, or the average per disc. And then the number of predators you hope to find in 100 sweeps to indicate control. So, blow this up. And this is our large nymphs. If you found, say, 10 large nymphs per disc, you'd be in that zone of uncertainty or around the threshold, and you'd be making a decision.
10 discs infested.
10 discs infested, sorry. Which translates to 33% infestation. And you're taking sweeps and if you found 2 or more big-eyed bugs or geochris, that would indicate control or suppression of whiteflies. Fewer than 2 would indicate a lack of control, or 2 or more would indicate control. So to put this in a more real-life scenario, I went out and sampled some fields around Maricopa yesterday. Went out, I took leaf turns. I found 9 leaves infested with adults and 9 leaf discs infested with large nymphs. So with that, I know that I'm in a situation where I'm around threshold and spray decision is needed. This would be a good time to take sweeps and look for predators and compare them to the whitefly population. So knowing that I've got 30% infestation, I'd want 1 or more big-eyed bugs, 1 or more pull-ups, or 3 or more pirate bugs. And just to be clear, each of these predators are independent of each other. They're independent, and they're indicators of the natural enemy community. So you wouldn't want 1 and 3. You just want 1 of these predators showing up at or above this level. Our whitefly infestation is also 30%. We'd want 2 or more crab spiders to indicate. So took some sweeps. I took 100 sweeps. I found 6 Lygus adults, no nymphs. And then I counted my predators in those sweeps as well. Out of 100 sweeps, I found 1 geochris, 5 pull-ups, 39 Orius, and 2 crab spiders. And I broke this down into 25 sweep reps. Because sometimes it's useful just to take 25 sweeps, or maybe just 50 sweeps. So we knew we needed one geochris. Is that right? Someone got the table.
You needed 1 geochris.
1 geochris. So I got that in our 100 sweeps. I actually got that in the first 25 sweeps, so that would have been sufficient to indicate control. Had 3 pull-ups in my first 25 sweeps, again showing control. But no crab spiders until the 3rd set of sweeps. So in that case, it would have been useful to continue sweeping. But this goes to show you, and this is a field that hadn't had any treatments. Whitefly populations were climbing. We didn't know how fast, but we knew that there was a large natural enemy community in place suppressing whiteflies, at least for the short term. So another way to think about this community of natural enemies is also an indicator of the population growth of whiteflies. If you've got, what you really want to know is whether whiteflies are climbing kind of slowly or if they're really taking off. And so one thing these predators do is help indicate, large natural enemy communities can indicate that whitefly populations should be suppressed. It doesn't mean they won't pass threshold, but it does give you a few more days or even another week between sampling to make that spray decision. Conversely, if all these predators had been absent, I would have known that we would have been in big trouble in this field. And those situations can occur if a broad spectrum's been put down, like Vivarin or Orthene, to knock out brown stink bugs. But the absence of a natural enemy community or a natural enemy community at very low levels would indicate that your whitefly population is really about to take off. In which case, you should consider advancing a spray before reaching threshold. So that's kind of the sum of the project, the research. We think this is a useful tool, again, really specifically in that area around threshold where a spray decision might be tricky or difficult to make. It shouldn't require any additional work except a few calculations or looking at this table. And it's not necessarily going to delay a whitefly spray in certain scenarios. It could advance a spray well beyond prior to threshold. So it's a tool that we think is useful, but we still do need to run some validations. We're running commercial validations with a couple of PCA cooperators. We want to get everyone's feedback as to how useful they think this tool will be or how readily adopted it might be.
But up next we'll have Bill McCloskey, and I just want to remind those watching online that you can download his handouts in the About section. There are 2 links to his handouts, so you can click on those and get those and follow along with him on his presentation.
Awesome, Shauna. Okay, Shauna asked me to talk about Morningglory control and some of our other summer weed issues. So we'll start off talking about Morningglory. It is one of our tougher to control annual species, always has been. This table in the first handout basically lists all the commonly used herbicides that we have registered in cotton here in Arizona, and I'll discuss just a few of them in terms of their efficacy for Morningglory control as well as other weeds. Really, in terms of the products that we have to work with, there has not been a whole lot of change for over a decade. Probably the newest technology up here on this table is the Liberty 280. Of course, to use that product, you've got to be growing cotton that has the Liberty Link trait. So in terms of a program for Morningglory control, there are a couple of key issues to think about. One is that the dinitroannulant herbicides, or the yellow herbicides, the DNAs, do provide some measure of suppression. If the expectation on the grower's part is that they're going to apply Prowl or Treflan pre-plant incorporated or in front of the mulcher and stop all Morningglory emergence, that's just not realistic. That seed emerges from anywhere from a half an inch to a couple inches deep. One thing that you'll typically see in cotton fields also, particularly on heavier ground, soil with a little more clay, is that it will crack and you will get emergence through those cracks, and where the seedling is able to avoid a lot of contact with the herbicide. But what we do see typically is about a 50% reduction in emergence, and those plants that do emerge stay smaller for a longer period of time early in the season. So it gives us a bigger window of application for our post-emergence herbicides. The herbicides that have efficacy on seedling Morningglory are Staple,
Liberty 280, and of course, glyphosate. Around the pretty varieties. One of the issues with Morningglory is that we really need to be timely with our post-emergence applications. We're going to have difficulty even with a 44-ounce application of Roundup Power Max or Original Max on anything larger than 4-leaf Morningglory. Once you get up to that 8-leaf stage, particularly when Morningglory starts to vine, a single application of glyphosate, even at the maximum label rate of 44 ounces, is not going to control those plants. You're going to be treating those same plants pretty much to lay-by. And so it really behooves you to be out there spraying 1 to 2 leaf Morningglory. One of the things that I often wonder about is what we see in our experiments is that when we plant cotton and we get emergence, the weeds that come up with the cotton pretty much come up in a single flush within days of those cotton plants. And there's very little benefit waiting, between 1 to 2 leaf cotton and 4 leaf cotton, or if you want to think about it in terms of Morningglory, 1 to 2 leaf Morningglory and 4 leaf Morningglory. The same plants are out there. And so it's really to the grower's benefit to make that application when those Morningglory seedlings are small, less than 4 leaf, because we will get them all with glyphosate. Now, when we look at our other selective post-emergence herbicides that have activity on Morningglory, the size issue is even more critical. With staple herbicide, we need to be treating basically 1 leaf Morningglory. If it gets beyond 2 leaf, staple at the maximum label rate of 3.8 fluid ounces per acre is not going to provide great control on those larger plants. We have the same kind of size limitation with Liberty 280 sprayed on Liberty Link trait containing cotton varieties. Those plants need to be small. Even at our 43-ounce rate of Liberty, basically you're just going to burn the leaves off of that stem, and it's going to take a second application to control those plants. So those 3 there at the top of the table are the selective chemistries we have available early in the season. After our cotton gets some size, our goal in Morningglory control early in the season is to get a height differential between our weed population and our cotton plants. So if we manage that first over-the-top application of herbicide well, we're going to achieve that height differential, and as our cotton continues to grow and gets into that 6 to 8 inch size, typically around the time when we're watering back and side dressing, applying nitrogen, starting to think about applying nitrogen fertilizer, or even larger, we can then go to post-directing our herbicides with a hooded sprayer or some sort of post-direct rig. If we're very timely, we can sometimes get enough contact on our weed population by continuing to go over the top on that second application. But with respect to Morningglory control, if you've got those seedlings that you've severely injured and they're within an inch or a couple of inches of that seed line, the only effective way you're going to get contact on them is to post-direct that second herbicide application. So something like a Red Ball 420 lay-by sprayer using the side nozzles, or a Wiley sprayer, or even a drop tube in the furrow with a double swivel can be effective in delivering the herbicide on those plants. Obviously, if you make a second application of Roundup, you've got pretty good selectivity between your post-emergence herbicide and your cotton. Same thing would be true of Staple and Liberty 280 on Liberty Link trait containing cotton varieties. And however, if we tank mix some of our older materials like Probotrine or Diuron, or use some of the relatively newer materials like Carfentrazone, which is AIM, or ET, which is Pyroflufen, then we need to be pretty precise in terms of where we put that spray pattern so that we don't cause excessive injury on our cotton plants. And then, of course, badly infested Morningglory fields are going to have to receive some sort of lay-by treatment in order to protect that cotton yield through to harvest. Morningglory is the one species that will emerge even after that cotton canopy closes and has the potential to grow up to the top of that canopy, depending on how competitive your cotton is and what the quality of your stand is in terms of skips and gaps in the canopy. And so we have a number of choices. Basically, lay-by in terms of herbicide labels is anything applied over about 15-inch tall cotton, 15 to 16 inches, 18 inches for ET. With some of our contact burn down type of chemistries down here at the bottom of the table, Goaltender, Ain, ET, and Chateau, we want to be treating only that barky tissue at the base of that cotton plant. We don't want to contact green tissue, green stem tissue, because we can partially girdle the cotton plant and cause some substantial injury. And the materials that have good residual activity are going to be things like diuron, chromatrine, Lay-by Pro, which is a premix of diuron and limuron. Goaltender has residual activity, and of course Chateau. At this point in the season, we need to be thinking about the residual characteristics of our lay-by herbicide application that we're using for Morningglory control, and we need to be thinking about what the next crop we're going to grow is. We have seen quite a bit of injury from lay-by rates of diuron and Goaltender on wheat following cotton, so those would not be good choices. In the last column over here, I've listed just a general overall categorization of the length of control. But if we look at other pages of this handout, I've gone through by chemical, by herbicide, and have listed some of the rotational restrictions that are applicable here in Arizona. So, for example, we can look at staple and see that if we're going to follow with field corn or grain sorghum, we've got a 10-month plant back interval. If we're coming back with wheat in the fall, it's only 4 months. Particularly if we do tillage. Here for diuron, we can see that for some of our grass crops, wheat would be in that category as well. There's a substantial plant back interval. Chromatrine is an interesting product. Caprol, the labels are actually fairly not all that different than diuron, but we know through experience growing fall planted wheat and barley that the activity in our soils is biological activity in our soils is pretty substantial, and we generally do not see problems following lay-by applications of chromatrine, Caprol or Cotton Pro on fall planted wheat.
So Bill, would that be true if you were going onto new ground that hadn't been in cotton for many years and no Caprol been used? I mean, are there greater risks under those conditions?
There's a little bit greater risk, but even so, sort of the general microbial population is able to degrade Caprol pretty rapidly. Gosh, there's so little new ground around here.
Yeah.
That is an interesting point, though, because there is data in the literature that shows that ground that has been treated with triazine herbicides does develop a microbial population that really chews that product up pretty fast. And the same is true to a lesser extent for the urea herbicides. If rotational restrictions are an issue, the ones that have the shortest plant back intervals are going to be Chateau, Lay-by Pro, which is the diuron-linuron combination, and Chromatron. Let's see what else is in there. Yeah. Okay, so now I want to switch gears and talk about the other handout, which I'm going to bring up here, but that's okay. Just to give you a brief update on where we stand with Palmer amaranth. We saw our first population of glyphosate-resistant Palmer amaranth in Buckeye in 2012. That's when it was brought to our attention. Given the nature of the infestation, it had been out there probably a couple of years prior to us getting called and going out to the field. Subsequently determined in greenhouse experiments that initial population was resistant to both glyphosate and also ALS inhibiting enzymes, staple LX was the one that I tested on that population initially. In fall of 2013, working with PCAs in Santan Valley, we discovered that there is Palmer amaranth out there that is resistant to the ALS inhibiting herbicides Raptor and Pursuit. Same enzyme system. It's inhibited by staple. The interesting thing about that Santan Valley population is that it is not resistant to glyphosate. It's still susceptible to glyphosate. So clearly a different population of Palmer than what exists in Buckeye. So most likely that population was independently developed by those growers out there. And then this year, in 2014, it's clear from talking to farmers and PCAs in Buckeye that glyphosate-resistant population in Buckeye has spread. It appears to be all the way from Goodyear out past Palo Verde. In that entire Buckeye area, there's a lot of areas out there where Palmer is not being controlled. We've got silage corn. We've got a lot of roadside areas that are reservoirs for that glyphosate resistance gene, and it's probably just going to become more widespread since it doesn't appear that we can control the Palmer amaranth populations across the landscape. I did sample some populations out there in 2013 and found resistance scattered around. We'll collect more samples this fall after the monsoon rains grow us a great crop of Palmer amaranth. On Monday, I went out to a field in Marana, and it's clear now that we have the glyphosate-resistant Palmer gene in the Marana area as well, sort of on the north side of Marana out by the Pinal County Air Park. If you see that big mound of dirt out there on the freeway, just south of there off of Luckett is where the plants are. And that's a really interesting situation. Very, very attentive grower. We're talking about just a few scattered plants in the field, growers on top of it, and is mechanically is cutting those plants out. In talking to him, the only two possibilities for the appearance of that gene, well, there's three really. I guess we could have developed it de novo, but it doesn't look like that. It looks like it came from somewhere else. The distribution of plants in the field kind of line up with where the irrigation water started on irrigation sets, indicating that it's a good possibility that seed floated in on irrigation water in the local canal system for the irrigation district. These fields that have this problem are kind of at the end of the line. So working upstream, there are some corn fields that have some pretty pretty hairy Palmer amaranth in them that's not being controlled, particularly along the perimeter. So we'll be sampling some plants in that general area this fall as well to see what we can come up, what we can find in those populations. So basically, these growers in Goodyear, for example, and here in Marana, you know, their options are they're going to have to use a dinitrotoluene herbicide to control as much palmer as they can early in the season. 1 or 2 timely cultivations will go a long way towards taking out those resistant individuals. And then in the seed line, where you have escaped from your Roundup applications, you're going to have to hand remove those plants. One of the questions that I get is, do we cut and remove all these plants from the field and bury them or put them in a burn pit and burn them? How concerned do we need to be about seed production on the females? And in looking at plants in that field, they're just starting to develop inflorescence, so we're probably 2 months away from mature seed. A plant like that can be cut and left in the field. It is going to dry out before any seed can develop on those inflorescences. Now, to be clear, we're talking about females that have an inflorescence spike, that main one at the top of the plant, that's small, just a few inches, 4 inches, not the 2-foot long panicles that you see on big individuals. If you've got a large inflorescence like that, you need to cut and remove that plant from that field, because those fleshy stems will mature seed that's close to being at maturity at the time that you cut. But something that's just got a very small inflorescence that's just getting started in the flowering cycle, those you can leave in the field. Our area conditions in between irrigations will allow those to dry up. Going forward, okay, so in this set of tables, I've just listed in Table 1 a general set of control options and tactics that can be used to manage cultivation, to manage herbicide-resistant weeds. Key is we want to use a diversity of tactics and not rely on any one chemical. Herbicide sequences are an excellent way to get a diversity of herbicide mode of actions in our cotton production system. One of the favorite sequences I like is using a dinitrotoluene herbicide, pre-plant incorporated or put out with the mulcher early in the season, followed by our post-emergence herbicides. Now, I understand that for a lot of our growers, they're using glyphosate not just to control palmer, but also for Morningglory and a spectrum of other weeds that they're dealing with. So although it would be ideal to limit the use of glyphosate in fields that have glyphosate-resistant palmer, that's not always possible. That means you're going to have some other tool out there to control those plants. And that can be mechanical cultivation. It can be using a different chemistry. It's one of the important things we need to determine in Marana is whether that population is also resistant to staple like the buckeye population. That field was custom harvested, so it wasn't picked with the grower's equipment. So there's a chance that seed came in on a harvester, but it's also possible that it came in the irrigation water. We don't know, but given that, so far, the populations in the state that have been resistant to glyphosate have also been resistant to staple. So my expectation is that's what we're going to find in Marana, and so that's what I would base my recommendations for control on. So, you know, obviously, when you discover this and you're already growing Roundup Ready cotton, you don't have the opportunity to go to Liberty 280. So basically, you're left with hand weeding and removal. But the following year, you better put a different technology in there and start moving away from spraying glyphosate. Acres of Liberty Link cotton in Georgia went from about 2% to about 50% in response to glyphosate resistance, because that gave them that other over-the-top post-emergence selective herbicide that they can put on palmer. It's a real challenge to make Liberty work on palmer. You've got to spray small plants, and I mean like 1 inch tall. to make that, to get control of palmer. Otherwise, those plants will survive, and you'll be retreating them with post-directed chemicals later, which does work. You can make that system work with a dinitro-aniline herbicide applied pre-plant incorporated. So one of the issues in terms of, well, how do we manage a diversity of herbicide products? One of the things that EPA is working with manufacturers on is putting a mechanism of action group number on the label, on the first page of the label, on the jug, so that you can readily determine, is this a different mode of action or just a different trade name for the same chemistry I have been using? So that's what this mode of action or mechanism of action group number is about. And I've listed here some of our common herbicides, both trade names and common names, along with a brief description of their mechanism of action and their group numbers, so that you can be thinking about using different modes of action in your cotton crop. And in the third table, I've just given some examples of the timing of use of different tactics for weed control. And of course, hand weeding is an option virtually all growth stages. And I want to finish up by making the point that sanitation here, in terms of dealing with herbicide-resistant palmer, is key. The appropriate response isn't to throw your hands up in the air and say, oh my God, there's nothing I can do. I'm just going to ignore the problem or hope that it goes away or what have you. So if you make a first application or first two applications of Roundup and you see escapes out there, don't delay. You need to stop seed production on your ground. So in any given patch of palmer, you're going to find about a third of those individuals are females. You can tell they're females by comparing the inflorescences of the male and the female plants. The males are kind of, particularly when they're young, if you take your hand and just run it up the inflorescence, they're smooth. The females got prickles. And as those inflorescences get larger, those bracts on the base of the flowers in the female plants get stiffer and sharper. And they were really sticky once they get some maturity to them. Also in the morning, on the males, you can see the yellow anther sacs, and if you shake that inflorescence, you'll see yellow pollen grains fly off in the air. One of the key points in dealing with this species is remembering that the resistance gene travels both in pollen and, of course, in seed. The seed's the important component in terms of what's going to be the future infestation on your land that you're farming. The pollen's important in terms of both having receptive females. You may have susceptible females in the field that are receiving resistant pollen, which is going to result in a problem the following year. But you can also be the source of pollen for infesting other nearby fields. So you need to remove all of them, in my opinion. But if you're gonna have a few escapes from the hand cultivation job, hopefully they're males, because they're not going to produce seed. So sanitation is key. It's my belief that unlike the Southeast, where they struggle to make residual herbicides, pre-plant incorporated herbicides work, or pre-emergence herbicides work in their system, the dinitro-anilines work great for us in our system. And a combination of doing that, using some of the other chemistries in cotton, we can manage glyphosate-resistant palmer. It's not the end of the world. Now, a crop that is really going to be a challenge with respect to palmer is alfalfa, because the only selective over-the-top chemistry we have in that crop are ALS-inhibiting herbicides, Raptor and Pursuit. If you've got this population of palmer that's got resistance to those two products, you have no way to control palmer amaranth in your alfalfa other than applying Prowl, H2O, either water running it, spraying it. And you've got to time those applications perfectly. That product's got to be out there before the palmer comes up. And then you've got to keep as you know, Carl, keep your berms clean.
Yeah, well,
Using the DNA in alfalfa and the frequent cutting goes a long way in the basins. It's the berms that really turn into the issue. Also, Carl's comment was that 2 applications, with that second one in alfalfa being in the, what would you call it, early summer. Same comment for the grasses, though. Will go a long way towards suppressing palmer in flood irrigated alfalfa. In terms of the future, if we get USDA APHIS deregulation of dicamba-resistant cotton, which potentially could happen this fall. I've talked to Monsanto folks, and there is a substantial amount of seed acreage being grown in Arizona right now. They will have a significant quantity of seed for the 2015 season. And dicamba is very effective on palmer. If we spray palmer a few inches tall, 4 inches and less, that's going to be a very effective post-emergence tool for us with respect to controlling glyphosate-resistant palmer amaranth.
not very likely to develop resistance to dicamba either.
There are weed populations that have resistance to dicamba. So I don't think the fact that we get that new technology means that we can move away from using a diversity of management tactics. Every new product that comes in has there's got to be some window of time where potentially you could use that product alone, but eventually there's going to be some weed out there that's going to develop a resistance strategy for that product. And we want to try to preserve these chemistries because dicamba is an old product. We're not getting a lot of new mechanisms of action in terms of weed management in the world. So it's probably been 20 plus years since a new mechanism of action was even discovered. So we're a long way from any kind of new product with a new mode of action. Seed producers are going to develop some other resistant herbicide technologies in cotton. But they're a number of years away. With respect to Morningglory control, tank mixes of dicamba and glyphosate are pretty awesome. They will really control significantly good-sized Morningglory that's vining. So that will improve our summer weed management.
But first I wanted to kind of review, these are from the current batch of cotton advisories this week. Just a little discussion of what, from a heat unit perspective, it looks like on the 2 weather stations that we're operating here up in Pinal County, one in Maricopa, one in Coolidge. This is the advisory that we issued on Monday for Maricopa, and you can see that earlier plantings should be at peak bloom. Later plantings are in the mode heading toward peak bloom. In general, the crop is about 3 to 5 days ahead of schedule. And from January 1, this has been a very warm overall year. We're 11 days ahead. So what you can see here is if our crop's only 3 to 5 days ahead, and from January 1 we're 11 days ahead, that kind of shows you how warm January, February, and March were this year. They were almost unprecedented in their warmth in this part of the world. So in general, that was good for getting the crop out of the ground. But it also maybe generates some challenges when it comes to things like insects and other things. Now, this is the Coolidge side of the interstate, so to speak, the east side. Here, the situation's a little different in that the crop, based on almost 30 years of data from that Coolidge station, is right about on the long-term normal. So it's been a little bit cooler on the east side. The annual accumulation is only about 6 days ahead, so there's about a 4 or 5 day difference between the 2 sides of the interstate, at least with respect to crop development and advisories. The Coolidge side does on an annual basis tend to run cooler, has since we put that station in many years ago. Part of it's where the station is sitting. It's a little bit of a poor drainage area and it's a little cold at night. Okay, now, this was something that we all annually await either with pleasure or hatred, depending on your point of view. In agriculture, generally we don't care much about this monsoon. But I want to go through very quickly a little detail on what the monsoon is and what's happening now and why this is off to a good start. On the left-hand side there is kind of a depiction of the weather patterns that we typically deal with in this part of the world. High pressure, this is a mandated high pressure system by atmospheric circulation physics, generally sits in this part of the latitude range. And it is responsible for clear skies, dry air, and very little precipitation most of the year. This is called a subtropical high. What happens with the subtropical high is that it shifts north and south with the sun angle, so to speak. So it will shift north in the summer and shift south in the winter. So on the north side of the highs is where we see our typical low pressure centers and winter storm track. And on the south side of the high is the true tropics. That's why Arizona is called the subtropics. We're kind of in between. So when we get into monsoon season, this high migrates from northern Mexico up toward the Four Corners region. And because air circulates around the high in a clockwise manner, it pulls in the hot, humid air off of the Gulf of Mexico and the Gulf of California, and that's what creates the monsoon season. So it is a true shift in air and airflow, and that's what creates it. Now, the thing that's been enhancing the monsoon the first couple weeks is not only did it shift very quickly and open the door up for all this moisture to come in, but on the south side of this is a series of circulations which we call inverted troughs or easterly waves. These are basically the storm systems of the tropics. We've had several of them wobbling along and wandering up into Arizona here in the first week, which has enhanced the precipitation substantially. That's equivalent, almost equivalent to a typical storm system you would see in the winter that will dip down in here, only these are tropical in nature, and they can really put out a lot of rain, as we've seen here in the last week. So, I did this slide at 5:30 this morning. This is the current precip totals as of midnight last night for the first 7 or 8 days of July. We're off to a tremendous start on precipitation. Coolidge is the low point out here. Paloma got some this morning after midnight. Parker is still dry and roll. Yuma's had a decent rain. Maricopa is the kingpin right now at 1.93, but they got beat up last night. Mojave Valley got a huge rain. In fact, got 2 inches in Mojave Valley in about 2 hours last night. And Del Wakamoto told me that it's just a mess. And
So after midnight?
no, before midnight.
Oh.
But the weather station in Mojave only picked up an inch, but one of his other stations picked up 2.
How much did they get from Maricopa last night?
Well, we had 1.15 in the previous storm, so it looks like we got another 0.8.
Yeah,
Oh, this is from as of this morning.
yeah, as of this
Yeah.
morning. Nothing after midnight.
Because I drove down Murphy Road today,
Yeah,
and
so.
it was swimming fast.
And Bill was complaining there about a week ago about if this is a wet summer, the Palmer's going to have all this moisture to. And to be honest with you, it's already looking pretty wet.
So Paul, you don't have Kansas settlement up there?
I removed the weather station.
Oh, you did?
Yeah, no funding.
No funding, no weather.
No funding, no weather. But you can see that Bowie has already had 1.2 inches down there. So this is pretty much region-wide in terms of the moisture coming in. And my guess is that Parker and these places just missed that mess over at Mojave last night. That was quite a storm. It went on about 3 hours. Okay, let's just talk a little bit about what the monsoon does. You all kind of have a sense of what happens here, but I thought I'd just show you graphically what really goes on when the monsoon comes into Arizona. This is a plot of this year's actual temperatures at the Maricopa Ag Center. And the red line is the maximum temperatures for the day, and the blue line is the minimum temperatures for the day. And this is the dew point, which is one of our indicators of monsoon arrival. And the monsoon started to come in about the 1st of July. You can see there's a very rapid increase in dew point. But even then, the dew point was slowly rising. And look at how the minimum temperatures come up. The real impact of the monsoon on general temperatures is that it increases the minimum temperature at night. You get clouds and humidity, you don't get radiative loss. The nights simply stay warmer. Because there's more clouds and humidity, the days tend to flatten out or even cool off slightly. So the result of the monsoon, and most everybody knows it, and everybody who's historically talked about cotton fruit shed will always say it's hot night temperatures that generate it. Well, it is in part driven by this because that's the most physical signal you see of the monsoon is an increasing night temperature. Now what's happens to the crop temperature? We have heat stress monitors sitting out all over the state this year. And this is from the Coolidge one sitting over on Sundance. And this graph shows the average air temperature for the day, the average crop temperature for the day, and here's our dew point again coming up through the system. So take a look at the average crop temperature, average air temperature for the day is kind of all over the place, up and down, but no direct trend. But look at this average crop temperature coming up as you get into increased levels of humidity. That is really what happens in this system is we simply have warmer nights, less ability to cool ourselves through evaporation, which is the way a plant helps cool itself during the day, and the average canopy temperature just continues to rise. When we get the right combination of high temperature and high humidity, we end up with a heat stress event. Now this is also off this monitor, and I wanted to show you the impact of humidity on these two things. Here is the crop in green and the air temperature in blue before and after the monsoon at night and during the daylight hours. So June 25 to 28 is a very dry period right before monsoon came in, and July 4th to 7th you've all experienced, so you know it's been quite humid. Thing to look at is at night the crop during the dry season 8.5 degrees cooler in those 4 days. In the daylight hours 15 degrees cooler because of the dry air and the evaporative effects. It's like an evaporative cooler. Look at post-monsoon event. Only cooler by 3.4 at night, only cooler by 8.8 during the day. These are actual measurements we've collected off this station this year. So you get an enhanced warming on both ends of the curve, but you don't see as much effect during the daylight hours because we tend to have a little cooler temperatures. But look at the impact of the humidity on the night temperatures and how quickly they come up. So the bottom line is what creates the stress problem with the crop is the humidity lessens the evaporation during the day and the radiational cooling at night because of clouds and humidity, and the whole crop temperature elevates as a result of that. And when it gets to a certain level, you end up in a heat stress event. Now the beauty of this year so far is I'm sure people are seeing shed. I've talked to some people seeing some shed. But when the monsoon came in, the temperatures really collapsed pretty quickly. Hasn't been exceptionally hot unless you go west of here. So we haven't had the crop temperatures in what I consider excessive levels now. Most of them have been level ones, which we define as crop temperatures in this kind of 82.5 to 86 degree range. You'll get light to moderate sheds sometimes of those, somewhat dependent on crop fruit retention. But the breeders, I believe, have done a much better job of dealing with that temperature range now, so there's a lot more tolerance in it. When we get good hot temperatures and humidity, we end up with a much greater canopy temperature. This is what generates the really serious heavy sheds where you see all the little baby bowls at the bottom of the irrigation run after you run an irrigation. Heat stress is kind of noted for knocking off very small bowls 2 to 3, 4 days after bloom. And if you get a serious heat stress event for an extended period of time, you can look in the flowers and you'll see this funny looking asynchronous design. There's very rarely any pollen here, but you also have an asynchronous male-female situation, and you end up with almost all infertile foals in that situation. Those that do form are hooked or beaked, or they're small, and are particularly unproductive. Now, heat stress is first and foremost driven by your primary air temperature and then the level of humidity that comes in. So you can see in the long-term climatology, Yuma is obviously worse, Parker's worse, Maricopa gets better, and when you get down to Marana, they don't worry about this much because they don't get these serious heat stress events very often, and they only get a few mild ones each summer. And you go down and talk to Marana growers, they don't seem to concern themselves much with this subject. This is just very quickly, these graphs I've used for about a decade. This is the seasonal dynamics of heat stress. Showing it generally, on average, starts about the 1st of July. It accumulates pretty consistently till about Labor Day and then flattens off and disappears. That's the shortening of the days and the drying of the air as the monsoon retreats. Here's the probability of heat stress, having at least a level 1 heat stress day in the Buckeye area, and you can see that in late July this peaks. About 80 to 90% of the days there in late July, early August have the potential to have heat stress. That's why historically we've always said it's very difficult to make cotton in August out here. You're just walking right into the worst part of the monsoon. The best time to make cotton is back in here, and if you're a late person and want to go late, on the back end of the season. First, level 2 heat stress typically comes in about the 13th of July here. The first heat stress events usually come in right around the 1st of July. So we're not abnormal on this year. And I would say we've been fairly fortunate in that the temperatures have been relatively mild, even though it's been incredibly humid. In fact, if you read the Weather Service jargon, our humidity levels are 150% of climatology, which means we're 50% above normal averages for this time of year. It is really steamy out there. Dew points in the 70s are not common in Arizona, and we've been in the 70s now for 4 or 5 days.
It was nice in Tucson yesterday, it was only like 90.
Yeah, well, when you get a lot of humidity like that, it ends up being very cloudy. Finally, just a comment or two. This is some work we did a couple years ago at Maricopa County, where we're looking at the yield trend. There's been a significant improvement in yields in the state over the last decade or two. And so in order to look at perhaps the impact of heat stress, we ran a trend line through here. And then we looked at the years when the data are above the trend line and years when the data are below the trend line. And when they're above, the heat stress levels are about half of the levels when you're below the trend line, suggesting again that heat stress does play a role in what you might consider the good and the bad years. It's not the only thing driving crop productivity, but it does have some impact out there. Okay.
Is there a physiological reason that occurs? Is it because the plant because it has less load on it after it has made a lot of bolls, and it's easier to cool off the portion of the plant that is still in the setting squares?
I think the physiological side of this, there's still a lot of arguing about the physiology of boll shed and heat stress. From our perspective, and this work is now 20 years old because we haven't had any funding to do it, is that there is a physiological mechanism in the boll that simply makes it sterile and just causes it to fall off when we have heat stress events. Some people will say it's a source-sink relationship. In other words, if you raise the temperature, the metabolism of the plant's running harder all night long, and it's burning up more carbon, and it has a lower physiological carbon balance, and then sheds fruit. And that's a legitimate argument. But the work that's been done in Mississippi, and some of the work we did in Tucson and growth chambers, where we altered the CO2 environment to increase photosynthetic capacity and other things, suggests that if you raise the temperature up, those bowls are going to fall no matter what the carbon status is. So, but there's a lot of arguing in the literature about what's going on here, and I think there's a little bit of truth in both of them. There's a bit of a carbon issue, and there's a bit of a physiological issue in that the reproductive structures simply don't develop correctly. And you see the same thing in corn. See the same thing in corn sometimes where the reproductive structures get asynchronous. That happens more often in corn with water stress, where, well, you also have pollen trouble with water stress, but you'll get asynchronous reproductive development. That's kind of what happens with severe heat. The male and female, you get all the pollen let go here, and here's the female part of the plant up here, and it's got to get up there, and it just simply doesn't. But a lot of times it just goes sterile. The pollen either doesn't form or doesn't get released. And we've seen both sides of that.
That probably is the bigger part of it, though, isn't it? The sterility issue?
Well, there's definitely a sterility issue. What I think happens is that after you have a heat stress event, sometimes you'll see pretty quick shed. And what we've seen there is that the pollen is there, but it won't release.
Right.
If you get a serious hard heat stress event, then what happens sometimes is you will damage the reproductive structures of the square. Squares come out and the flowers are deformed, and you get these funny shaped bowls and asynchronous reproductive structures. So I think there's two things going on here. One of them is release of pollen, and two is just either total sterility or reproductive disorganization, if you want to call it that. It's a subject that continues to get a little bit of study in the humid South, but not the way I'd like to see it looked at. They don't believe they have it in the humid South. They have the same problem. It's so humid they don't realize it.
Yeah, but Paul, we can have some super hot days in June, and you don't see the same degree of reproductive deformity as you do if that same temperature occurs in July. So it's just not simply
But they're getting
temperature.
No,
over the night, though.
it's the humidity effect. As I was trying to show it, the crop is just running so much cooler in that dry air. Especially at night, these crops drop into the 50s. If you look at these foliage temperatures in June, sometimes the crop is 58, 59, and the night's 70 degrees. Because it's so dry, it just loses so much energy that crop surface gets quite cold. So the air temperature really isn't the big factor in driving this. It's the air temperature plus the humidity. Okay, we are running some heat stress monitors out in the state. And I have a handout here which we can pass out on how to get at this. I've given this presentation a couple other places, but bottom line is if you get on this URL, and it's on the handout, you can get plots of the canopy temperature as measured with infrared thermometers out in these cotton fields, and we're also measuring some additional parameters. And what we've done is made a three-color graph there, one green being no stress, yellow being level 1 stress, and red being level 2 stress. And here's the Paloma one for this year, and here's the one from Coolidge. Coolidge, we've been nipping right at the edge, but we haven't really had a serious bad day over here. We've had we've had one pretty serious day down here in Paloma, but most of the rest of them have been sitting in the level 1. So take a look at these. We're doing this work in part.
You had one, what was that first dated the first time it peaked into the level 2? Is
July 4th.
to the left there is.
Oh, this was before the canopy got covered. This was when you put these out early, the crop's not fully covered. You pick up some ground temperature.
Oh.
So this is back in mid-June when we put it out, and the crop was only about a foot tall, and I couldn't get the infrared thermometer not to see the ground. The ground gets just hot as blazes during the day. So, but this you can see it after the canopy gets developed. It sits down here in the green zone most of the time at that point. So, and we have been doing some model evaluation. Most of you know we've been putting out a heat stress report for God knows how long. I don't know, maybe 15 years. One of the things we're doing with these monitors is to check whether the model's any good, and in most cases where we're checking, we're getting pretty good agreement with what the model temperature is that comes off our reports off ASMET. and the measured temperatures with the infrared thermometers. So I think the model's pretty good, but we will probably make some adjustments to it at the conclusion of these analyses. Okay, what's the impact of heat stress? Well, most of the time it's fruit shed. And I'm just going to point out an old graph from Silvertooth. I had an email exchange with him last night, but it had nothing to do with this. But this is a graph that Jeffy put out in the Cotton Report many, many years ago, and it has 3 scenarios. One with high fruit retention, one with poor fruit, normal fruit retention, one with declining fruit retention. And it shows, and this is the percent fruit retention, and on the right is the height to node ratio. And the point I wanted to point out, or the line I want you to focus on, is that number 2 line, which is one that's trending from near normal fruit retention to below normal fruit retention, and then look at what happens to the height of the plant. And this is the number 1 issue that I hear from growers when they start to get fruit retention. The plant goes vegetative. Starts to go vegetative, starts to take off. A lot of people will use these heat stress indicators as a timing mechanism for picks. Some people wait till the event occurs to put picks on to try to prevent this rather large increase in height to node ratio and vegetative growth. The other aspects that we've seen over the years is sometimes vegetative plants cutouts delayed because the fruit retention is low, so the plant tends to slow it on its maturity, and that can impact the maturity of the plant as well as defoliation. If you do not utilize all the nitrogen that you put in the ground for that crop, and you get near the end of the year and you got a high nitrogen balance in the leaves, that tends to result in an increase in the struggle of getting the crop to defoliate correctly. Okay, let's go on to the weather forecast, because this is kind of an interesting one. I've been saying this all year. The Weather Service came out very early this year and said, we're going to bias the monsoon wet. They don't do that very often. In fact, I've been here 30 years. It's my 30th growing season. And this is only the 2nd time I've seen them forecast a wet bias monsoon. Normally, they just don't think they can forecast it. But this forecast right here, which is a precipitation bias forecast for July, has been on the books now for about 3 months. And right now they look like geniuses. This monsoon has come in on the 1st of July. We've had significant wet weather and very, very humid conditions. So they're forecasting a slight warm bias on the western side of the state, normal through central and eastern Arizona, and a generally wet bias for July, with the exception of maybe the southern edge of Arizona along the edge down toward Yuma. That's for July. Now, when you combine July and September, their 90-day forecast, things don't change much here on the temperature side. Western Arizona, slight warm bias. The rest of the state, kind of in the normal. You see the same wet bias throughout, maybe not quite as far south in Arizona, but certainly cutting across the central part of the state. So they're still suggesting this is going to be a wet-oriented monsoon. Now, here's the other issue that's coming to the fore, and we aren't quite sure where this is going to end up. There is a developing sea surface warmth going on in the tropical Pacific. This is South America, this is Indonesia, and this is the tropical Pacific. And those are, it's about a week old, but that's the bias or anomalies, which are above normal tendencies for sea surface temperature in the tropical Pacific. Now, this is a signature for El Nino to develop when everything along the tropical Pacific warms above normal. Normally, we end up with an El Nino event, which creates and can create exceedingly wet winter events out here. So this has been developing. It's been in the picture for quite some time. It has not been declared an official El Nino yet, partly because of the technical issues related to it. But as a result of that El Nino event coming along, we now have a very interesting fall and winter forecast. the fall forecast is for a rare event out here. They have blue out there, which is a cool bias. They also have a wet bias. So for the period October through December, they're now saying bias toward cooler weather and bias toward wetter weather. We haven't had a fall forecast for wetness in a heck of a long time, and we haven't seen this in at least a decade. So I don't know quite what they're saying, but what we feel they're saying is that this El Nino development and warm sea surface temperature may in fact produce a wetter than normal fall, which when you get the entire desert wet, you're going to end up with evaporatively cooled systems, and you're not going to be as warm as normal. Other point I wanted to make out from this graph, this is Mexico. Look at the warmth in here, and the warmth goes all the way up to the Pacific Northwest, and this is something we haven't seen in a decade or two.
That goes all the way up to like Seattle?
Yeah.
Holy cow.
There's very warm water off the Pacific coast, and we think one of the reasons that the wet bias is in the forecast for the fall is the concern about increased tropical/hurricane activity out in the Pacific, of which some of the moisture and or the remnants of those storms may tend to move up into the southwest and create this wetter than normal forecast. Because these waters are really warm, and they may be one of the reasons the humidity right now is so bloody high, because the air is coming off of these very warm waters. So that's the forecast, but this is also a forecast of an El Nino event. Southern tier of the states, wetter than normal. Now when you go to the back half of the so-called winter, January to March, the coolness disappears, but it remains across the rest of the southern tier. But you can see we still have a wet bias all along here, so this is all very representative of what they think is a El Nino forecast for the winter months. So I think the take-home message is here, the fall right now is being forecast to be cool and perhaps wet. So in planning on cotton, it's something to think about. We haven't seen a cool, wet fall in a long time, and if this comes to fruition, it does impact cotton, completion of cotton crop, as well as getting the darn stuff harvested and the groundwork before it gets too wet. So we will know a lot more about this in about 6 weeks. This El Nino has to get locked in pretty quickly, or we're going to start to think that maybe this could end up being a dud. Let me just show you very quickly what the fall-winter metrics are on El Nino. This is for the Central Arizona climate zone right there at Pinal, Maricopa County. This is the fall and winter precipitation when you have La Nina, which is the dry side of this El Nino cycle. When it's in the middle and when we have an El Nino, and you can see that as you go up toward the El Nino category, the winter precipitation goes up. These green and red dashed lines are the long-term averages. So when you get into El Nino, you typically have above normal winter precipitation. But there's a huge but in this answer, and it is that strength of the El Nino matters, and that's why everybody's on the edge about what's going to happen this winter. If you have a neutral condition, this is your precipitation patterns. If you have a weak or moderate El Nino, you can see you're just barely above normal. But when you get a strong one, then you have a much better tendency to be very wet. When you have a strong El Nino, it's about a 70 to 80% chance of above normal winter precip. You get into these weak to moderate ones, and it gets more and more like a coin toss. You can get very wet, you can also get a dud. So it's very important. Also, when this thing locks in, and we'd like to see it in place if you're going to have a wet winter by late summer. Finally, there's the history of strong El Ninos in the south central climate zone. You can see every one of them above normal, but some of them incredible. A lot of us lived through these two. These were incredible events, very, very wet events. But these go all the way back to some of the great wet events, 82, 83, even into the 50s. You can see what happens when we have a strong El Nino year. Here's what happens when you get a moderate one. You get some years that are incredibly wet, some years that seem to be duds. So that's why there's a little bit of waffling on the forecast, but everybody's providing the warning that it's possibly coming to see us this year. Finally, I won't go into this, I've gone into this, and my time is up. We do just want to advertise and have a handout here. The ASMET data is available in real time, and so if you click on that button on the ASMET website, you can get all of the data in real time updated every 15 minutes. And that's how I know whether it rained last night or not. I can look all around the state,
Do the
find out.
graphs that are on that current weather, you have some graphs like of the last 24 hours on there.
Yeah.
Does that include, is rainfall one of the ones you?
No, we're probably gonna put rainfall on cumulative rainfall. Yeah, we've talked about putting it.
That'd
You
be
know,
nice.
90% of the time, 99% of the time, it's going to be a flat line 0. But
Right.
yeah, we've thought about
Yeah,
that.
but I can look at it, oh, not going to Mac today.
Yeah. So anyway, and as Bill said, if you click on the location-specific tabs, you can look at what's happened over the past 24 hours. So with that, I'm going to either take questions or sit down. Yeah, Bill?
I have one question. So Paul, typically we get a break between the monsoon and the fall weather. Is there a risk that if this El Nino locks in, we won't get that break this summer? It could just rain right on through trying to defoliate?
The question Bill was asked is, normally we have a break between the end of monsoon and the beginning of what we might consider winter or the rainy season. And that is statistically very true. His concern was, could this end up just being a continuously wet period in the fall? That's kind of what this forecast is suggesting, is that we might not have that, what typically is 6 to 8 weeks in there between Labor Day and maybe end of October when it's generally speaking dry. That's what they're suggesting here, is that that's going to be maybe a more fractured period in terms of having some storm events in between there, which leads to your question, is that going to impact finishing the crop as well as harvesting it? And I'd say that type of forecast is a bit frightening to me with respect to both those issues. It could raise challenges with terminating the crop. It could also raise challenges with picking it. And not to mention the post-tillage, which is often where we get caught in these El Nino years, is we get the crop out, but then we can't get the groundwork. So I would say, Bill, that these forecasts get updated next week, I think the 17th of July, and when they get updated, that's when we'll have a little bit better feel for whether they still believe this wet fall scenario is in place. But right now, it looks like they're biased that way. Yes?
Is there any linkage between incidents of hail and that El Nino?
Question is whether there's any linkage between hail and El Nino. Not that I know of. In fact, one of the things that's very interesting about this forecast this summer is typically when an El Nino is developing, the monsoon is weak. That's kind of been it's a weak but realistic statistical relationship that the Weather Service believes in. And they were shocked when they saw the wet bias come out, seeing the El Nino developing. But I'm looking here 7-8 days into the season so far, and I'm saying, well, they haven't missed the first 7 or 8 days. This has been a pretty good.
Well, we've been having some pretty stiff winds, too.
Yeah. So, but we did get some hail out of some of these storms. I don't know whether you did, Carl, but there's been some hail bouncing around here in places. I haven't heard of any serious damage. Yeah.
I haven't had any yet.
Yeah. You don't want that.
So, I'm sure you're all familiar with what plant growth regulators are. There's 2 types. We have inhibitors and promoters. So, here in Arizona, we're often dealing with the growth inhibitors. And so, what the main purpose of those growth inhibitors is to obviously slow the growth and guide the plant to a bloom period where we have developing bulbs. So the types of plant growth regulators or PGRs that we have. We have the primary one, mepiquat chloride. Then there's mepiquat bentaborate. And actually, I think there's only one of those commercially available. And then, of course, you have all your mixes. The idea behind the mixes is that they can help improve yields and also be more efficient. Some people believe it works, some doesn't. I guess it's kind of dependent on what variety you have planted, where you're at, and that type of information. So why do we use the PGRs? So when we talk about our farming, we should have an ideal plant height at the end of the season that we're trying to go for, and we want it to be sufficient that it supports optimal yields, and also it helps with the ease of management and harvest. When we have excessively tall plants, it can result in delayed maturity. We can have problems with our insecticide and defoliation efficiency. We can see lodging issues, bull rot, problems with our harvest, and also potential for yield loss and quality issues. So I guess the biggest questions when we talk about PGRs is when you want to apply those. And it's important to remember that the effect of the mepiquat is a function of plant size and is applied of plant size and the rate, and so it only affects new growth. So has no effects on portions of the plants that stop growing. So what this means is that if you were to put a given rate of mepiquat on a small plant at first square, it would have a greater effect than if you were to apply the same rate at a plant that is in late bloom. Oh, sorry. So when we talk about vegetative growth of the plant, and we're usually looking at an application window for these PGRs from first square to cutout. So that's when we're actually going to see a benefit from these PGRs, is, again, at this time when it's going to be growing. So Paul had mentioned height to node ratio and fruit retention, and I kind of wanted to go over that with you guys on how to do that. I know Randy Morton's put out some videos and stuff, but it never hurts to go over that again. So I'm going to grab a plant that we got this morning out in the field. A few plants, and hopefully do some plant mapping for you guys. So you would measure the plant height from the soil. These have the roots on there, so I wouldn't take that height measurement. But and I would also probably have a better ruler than I do now. But I would say this plant is about 25 inches, 26 inches high. And I just threw that down. So we'd record that, and then we wanted to measure the nodes. So we'd start down here. We have our primary ones here, so we don't count those, and then we start counting. And they alternate up this down. So we'd have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So we have 19 nodes. So to figure out our height to node ratio, That's just going to be dividing the plant height, which we had was,
0.4,
oh, thank you. So
1.36.
1.36, thanks. My math is not very good. So our height to node ratio is 1.36. And then we can, let's go ahead and talk about fruit retention. So when we talk about fruit retention, we can, using the same plants, we have their plant height already, and we want to determine the number of fruiting stems versus vegetative stems. So the first fruiting stem is where you would see a bowl in that first position. So this one wouldn't have one in the first position. There's no bowl there. This one. So our first fruiting stem would be 1, 2, 3, 4, 5, 6. At 6. So we would record that. And then we would want to see how many sites have been aborted. So when we look at that, we want to look at the first 2 positions on the plant to determine whether it has either aborted or that there's a fruit. So first position, there's a fruit, 2 sites, no aborts, no aborts. Looks like we have 1 aborted. So from our sites, so there's 19 nodes, so 6 of those were vegetative, so the total of 13 fruiting. So the total potential fruiting sites that we would have would be 26. Out of the 26, we only found 1 abort, so 25. So we actually have a pretty high fruit retention for those. And I know I went through that fast, but there's a handout, and I will also be posting those later online for you to look at. So as Paul had brought this slide up earlier when he was talking about PGRs. So over here we have the fruit percent fruit retention. We have our high thresholds, normal, low, and then our height to node ratio. We have a high, average, and low. And so when we make these plant growth regulator decisions, we can use those to determine if we would see a benefit of applying these PGRs. So this is something Randy Norton had put together. This was done with a lot of work from the Cotton Cooperative with cotton specialists all across the state. And this is more localized data though. So what they did was we have this matrix that he created. And so if you have, you're looking at a percent fruit retention, and let's say you have a higher threshold, and then your height to node ratio, where it falls at along these lines, you can use the matrix to see if you would get a yield response if you applied PGRs. So let's say that since we had a pretty high fruit retention on these plants that we got, let's say we're along this A line here, and Peter was saying earlier that a height to node ratio was around 1.36, and I happen to know that these plants were planted around the beginning of April, so we're talking about a 2,000 heat unit accumulation. So we're looking between this A-B area here for the height to node ratio. So if we follow the matrix, we go to the fruit retention. So we're on A, and we're kind of in the middle of this A-B line here. So if we were to put plant growth regulator down, there is a pretty good chance, very high chance that we would see a yield response from that. So this was, as I said, this was information that was put together from studies done over a decade, but I guess they stopped in 1999, but I think the information's still pretty good. So, when we see a positive yield response from these PGR applications, we had We had a height to no ratio that was high, and then our fruit retention, if we had 61% or below 39%, and we were talking about the matrix here, we would see a positive yield response. Most often, though they're not, we see no yield response when we apply PGR, but again, going back to the reason why we put down these plant growth regulators is to regulate height and help us later on when we're defoliating, harvest, applying insecticides, and all of that. So how much do we want to apply? Obviously, we're going to want to follow legal rules, but general recommendations are putting down 8 to 12 ounces at first bloom. And then higher treatments following that. So there's no point in putting anything down after the cotton has reached 5 nodes above white flower. Some people believe that if you put it on after that, what you do is you stop the growth and it allows the bolls to mature and develop. But there hasn't been any consistent data that proves that to be true.